opening and closing of stomata, some basic terminologies of turgidity and flexidity in plant cells, and then the what is so unique about leaf, what is the structure of leaf, and then we came then we returned to this diagram in which we said that there are different portions of the leaf tissue that are used to leaf tissue actually, which in which some of the cells do not possess the chlorophyll, and some of the cells possess the chlorophyll. Some of the cells possess more chlorophyll, some of the cells possess less chlorophyll. So all of these things were discussed, and the cuticle is also present on the surface of the leaf. So what is yet to discuss? The word equation and balance equation. Actually, we didn't we didn't reach the process of photosynthesis. We were discussing this question. We were in the last lesson we were discussing about this question, that what is so unique about the leaf. So first of all, the leaf um, possesses the chlorophyll in the palisade and spongy mesophyll. And there is another type of cell in which the chlorophyll is present. Those are guard cells. And we did discuss in the last lesson that the guard cells start photosynthesizing in the morning and they become hypertonic, and then suddenly they absorb the water and become turgid and open the stomata. So this was a detailed discussion in the last lecture. So let's move forward. Palisade and spongy mesophyll, these are two primary cells which are containing the chlorophyll. And if you refer to the structure of the leaf, you would certainly say that the layer that is present above, this layer that is present in the upper side, actually this layer is directly facing the light. And just below the upper epidermis, if you refer to the structure of the leaf, you will come to know that just below the upper epidermis we have palisade mesophyll. So for example, I was in a past paper session with O-level class, and they asked me that, sir, there is a question in the past papers that what is the distribution, what is the effect of light on the distribution of chlorophyll in plant cells? Let me repeat the question. What is the effect of light on the distribution of chlorophyll in plant cell? So I told them that this effect is described in the book as the palisade mesophyll contains more chlorophyll. Why? Because the light that is coming from the sun actually falls first, maximum light falls first on the upper epidermis. And although upper epidermis does not possess the chlorophyll, the light penetrates into the upper epidermis and falls on the palisade mesophyll first. And then after penetrating all through the palisade mesophyll, it comes into the spongy mesophyll. So spongy mesophyll would be possessing lesser chlorophyll than the palisade. So this is actually about the distribution of the chlorophyll in the leaf. And why the chlorophyll is so important? Because the chlorophyll is a machinery. It is a molecule. It is a molecule that is the token of converting the radiation energy into the chemical energy. Actually, it is a token of, you may say, it is a token of confirmation that if any of the cell possess the chlorophyll, it has the capability to convert the radiation energy coming from the sun into the chemical energy. Did I tell you about this conversion? Yes. So
Yes.
this is a speciality of the chlorophyll molecule. So now what there can be a question that if we say that the chlorophyll is present in palisade mesophyll and spongy mesophyll, then where is the, or what is the location of the chlorophyll? because it can be asked that although chlorophyll is a very important molecule, still it is a molecule. So what is the location? What is the exact position? What is the exact location? So let's draw the structure of a plant cell, and then we would be discussing about, yes, you want to ask something?
No, I wanted to answer your question.
Oh, okay. Yes, go on, go on.
It is present in the cytoplasm inside the plant cells.
Not the chlorophyll. This is why we must be having an idea of the presence of the chlorophyll. For example, this is the vacuole of the plant. This is the vacuole. And now I'm going to describe the position of the nucleus, which is very important. Position of the nucleus is on the side. But what is the important substance? What is the important part that actually relates to the presence of the chlorophyll? That is actually the chloroplast. Chloroplast is actually the indication of the presence of chlorophyll. What I want to say that if you are talking about the plant cell, then never say that chlorophyll is present in the cytoplasm. You have to answer it in this way, that the chlorophyll is present in the chloroplast, which is present in the cytoplasm. No doubt this is cytoplasm. But first you have to understand that chloroplast is an organelle, is an organelle. Chloroplast is an organelle.
Sir, I'm unable to hear you.
Oh, I'm sorry. Is it right now?
Yes, it's okay.
Okay. Chloroplast is actually the organelle. Now, let me give you a very slight idea about the organelles. Organelles are the part of the cells which separate their reaction from the cytoplasm with the help of the membrane around it. For example, if you talk about the chloroplast, chloroplast is an organ that possess the double membrane, and all of the photosynthesis occurs inside. All of the photosynthesis occurs inside. So it means that the process of photosynthesis is separated by the processes of cytoplasm through the double membrane in the chloroplast. So this is the actually the this is one of the purposes of the organelles, that their membrane separate their processes from the process occurring in the cytoplasm. So this is the location, or this is the position of the chloroplast. And how does the chloroplast carry out this reaction? Let me tell you a bit further. Actually, chloroplast, the rule of the photosynthesis. Now, let me make it sure again that I should be telling you about basic theme of the photosynthesis. The rule of the photosynthesis is to combine energy poor inorganic substances to create or to synthesize or to synthesize energy rich organic compounds. Rich organic compounds. This is actually the theme of the photosynthesis. So you can see that there are two inorganic compounds. Both of the compounds, be very clear about it. Plant only creates the food if it is provided with the right compounds. For example, if you provide it with magnesium, if you provide it with sodium, if you provide it with water, if you provide it with carbon dioxide, if you provide it with ATP, etc., 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 if you provide right inorganic compounds to the plants, only and only then the plant is able to perform the photosynthesis. What does it mean? That if you cut off the supply of the chloroplast, oh sorry, if you cut off the supply of the carbon dioxide, if you cut off the supply of the water, even you would be needing a suitable temperature. You would be needing a suitable temperature. So it means that you can, the right things must be supplied to the plants. So let's have a look. The inorganic compounds, carbon dioxide from the air and water from the soil, these would be provided to the plants, and light from the, obviously the ultimate energy, ultimate source of light is the sun. So light from the sun and the chlorophyll that is possessed by the plant, all of these would help the plant to perform the organic compound, which is the glucose. And in this process, the plant releases the energy. Focus on the point that in this process, the organic compound that has been made is composed of three basic elements, CH and O. Now, this compound would be used to create further carbohydrates, fats, because the basic elements that are present in carbohydrates and fats are common, CHO, similarly CHO. There may be some of other elements are present, but the basic elements of the carbohydrates and fats are CHO and many other molecules, like we can use the CHO from the glucose to make the protein, because the CHO is also common in the protein as similar as fats, but it also requires the nitrogen because amino acids contain an amine group. So this glucose is used to create the other compounds as well, but this is a general process. Although it is a very lengthy process, but we are not meant to describe this process as as much lengthy as it is. We are actually destined to tell to know, to understand that if we variate the amount of carbon dioxide, if we variate the amount of light, and if we variate the amount of temperature, what would happen to the photosynthesis in that case? Didn't I tell you that? Didn't I tell you in this lesson that if the right things are properly provided, then the photosynthesis would occur. But if the right things are not properly provided, for example, you have provided the light properly, you have provided the water properly, but the carbon dioxide level is still short. So the photosynthesis would not occur properly. So what does it mean? It means that this reaction can be altered. This reaction, let me write it down. The photosynthesis reaction, the photosynthesis reaction can be altered by varying the amounts of CO2, water, CO2, water, and temperature. Oh, sorry. Not water, not water. Although the water is included, but we are not supposed to study the water according to our curriculum. Carbon dioxide, light, and chlorophyll. Excuse me again. Carbon dioxide, light, and temperature. So this process can be, all of these things must be supplied in the right amount for the proper level of photosynthesis in plants. So these, what do we call these factors, which are you may say that which can alter the rate, the photosynthesis reaction rate, I must write, the photosynthesis reaction rate can be altered. What do we call these factors that can alter the reaction rate? Can you answer this?
Factors affecting the rate of photosynthesis.
Okay, let me ask it generally, that what do we call those factors which can increase or decrease the rate of any reaction?
Abiotic factors.
Certainly, but there is another name. These li limiting reactants or limiting factors. These are limiting factors that can influence on the rate of photosynthesis or the rate of any reaction. For example, if you are dehydrated, then it means that your body is not performing the reactions properly. It means the water can be limiting factor for your body. The increase and decrease in the amount of water, the variation in the amount of water can affect on the reactions that are occurring inside your body. So this is a very important one, that we must know that limiting factors are those factors which, if not provided properly, then these can severely affect any reaction. We will be understanding these graphs today. So let me tell you, let's have a discussion about when are these factors altered. For example, when the light is altered, just be on the natural condition, not artificial, not man-made condition. No, just be on the natural condition and tell me what is the time of alteration in the level of light.
At the nighttime and at times of rainy seasons, because the
Uh,
clouds cover the sky.
yes, you also can say that the light variates around 24 hours. Why am I saying this? Because the angle of the sun changes in every hour. So it means that it can be changed around 24 hours, but we must know that there are the very major alteration or very major change in light for photosynthesis is in the evening and in the night, obviously. These are two major changes. In both of these time, and let's include the day, because we are not discussing about the decreasing intensity of light. We are just saying the changing intensity of light. So obviously we are including the day and the dawn as well. So these are the four times in which light can variate. And obviously when the light variates, it would be affecting the level of photosynthesis, obviously. So this is very important. It means that the rate of photosynthesis does not remain the same even during the day. It is different at the dawn, then it is different during the day, it is different in the afternoon, it is different in the evening, it is different at the sunset, and it is very different after sunset. So this is a whole category of light shades. If we observe our 24-hour clock, then it is a complete shades of the light. So we can assume these four times: evening, night, day, and dawn. So let's come to the temperature. Obviously, temperature also changes. For example, we can also include all four of these in the category of temperature. The temperature is different at dawn. It is different at 12 o'clock. It is different in the afternoon. Or let's write the day. It is different in the evening. It is different at the sunset. Uh, let's not confuse ourselves with the sunset, because at the sunset, even at the sunset, the photosynthesis is not occurring, and the temperature would obviously very much different at the night. So these are also the times when the temperature variates, and along with these variations, the rate of photosynthesis also variates. The optimum temperature for the plant to perform the photosynthesis is 35 degrees centigrade almost. 35 to 37, you can write 35, you can write 37. This is the optimum temperature. And also, you must know that this is not the confirmed or uh, constant temperature, because in many regions, the photosynthesis is performed very efficiently at 20 even. 20 degrees centigrade, 25 degrees centigrade, 30 degrees centigrade, depends on the species. But as far as our areas are concerned, it is around 35 to 40, you may say, the optimum temperature is. And the next thing is carbon dioxide. The question is, when does the carbon dioxide variate? A very important question. Can you answer this?
It's higher at the nighttime rather than in the day.
Well done. Why it is higher in the nighttime?
Because in the daytime, plants usually produce oxygen in a higher amount, and at the nighttime, they usually diffuse off carbon dioxide.
Okay, let me write an expression here. During the day, photosynthesis, which is releasing the O2, is at a very has is being performed at a very higher rate compared to the respiration, which releases the carbon dioxide. In which of the um organelles the respiration occurs?
The stomata.
Organelles, not the cells.
Oh. Um,
The respiration occurs in the mitochondria. Have we discussed the respiration?
I don't think we discussed it in terms of plants.
Oh, no, 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 no. Don't ever confuse the respiration in humans and respiration in plants. It is common everywhere. It is even common in bacteria.
Oh, okay.
It never changes. The respiration is the common concept in every cell. Regardless, it relates to the plants, it relates to humans, it relates to animals, it relates to insects, it relates to bacteria. It is common in all the organisms. So it is common for to produce the energy. Now, coming back to the point, we can write the expression that during the day, the photosynthesis is performed at very much higher rate than respiration. So it means uh, the plants is right now currently releasing the O2 into the environment because the O2 oxygen is more than the need. It is more than the need. The oxygen is more than the need of the plant. So oxygen actually is released because the plant does not need it. It is just like the scrapped material. It is just like we say in Urdu, bacha kucha khana. It's just like that. It's just like leftovers. That plants, when it does not need any oxygen, it just throw it away through the stomata. So it means that it is actually the leftover of the plant. So during the day, it occurs. And when we talk about the night, then we can certainly say that photosynthesis obviously is not occurring because there would be no light. Is none The reaction of photosynthesis is nil as compared to the respiration because the respiration is still occurring. The respiration does not need any stimulus of light. The respiration does not need any stimulus of any other thing. It actually happens. It actually is performed by every plant cell whenever it needs. For example, the plant needs the respiration, the energy from the respiration during active transport. In many other, in protein production, sometimes the plant requires the energy from the respiration during protein formation, during mitosis. So it means that respiration occurs in the whole day. It is constant in the whole day, at night and in the evening, almost constant. Not as much as the respiration does not occur at the rate it occurs during the day, but still it occurs 24 hours. So coming back to the point, now what would happen? Now the carbon dioxide level is too much in plant. So plant would release the CO2 into the environment. Now coming back to another point, is it only the plant that is releasing the carbon dioxide?
No.
All organisms. So it means that at night, all organisms, the carbon dioxide from all organisms would increase the level of carbon dioxide during the night. And during the day, the photosynthesis level would be this much higher that the carbon dioxide exhaled by all living organisms, even that carbon dioxide would be insufficient for photosynthesis to occur at a higher rate. Let me repeat my sentence. All of the carbon dioxide released from all of the organisms is insufficient for the plants to perform the photosynthesis at an ideal rate. So the level of photosynthesis is this much higher during the day. Okay, let me tell you one more thing. Let me tell you one more thing, which would actually um, either it would help you in your concept, or you would get confused. But you would not get confused. Actually, I'm not telling you due to this. Actually, I'm telling you because this type of questions I have seen in your past papers, and this type of question come in the O levels, A levels many times. Okay, the plants perform respiration. The respiration means you are breaking the organic compound into carbon dioxide and water. The water would be used in something. What would be the fate of carbon dioxide? Either it would be released out of the stomata, but only at night. During the day, that CO2 would be given to the chloroplast. The CO2 would not leave the stomata, rather it would be given to the chloroplast. Why it would be given to the chloroplast? Because chloroplast is performing the opposite reaction to the respiration, and that is called as photosynthesis. So this oxygen, it can have two fates, and it does have two fates. It can be released in the outer environment through stomata, and it can be given to the mitochondria for respiration, because we know that respiration does not occur in mitochondria, occur without oxygen. What is the process that occurs during without oxygen?
Anaerobic respiration.
Yes, or fermentation. At day, or we can say during the day, photosynthesis is higher, much higher. During the night, the respiration is higher. But what would happen when it is evening? The rate of photosynthesis at a certain time would become equal to the rate of respiration. What would that mean? That there would be no release of carbon dioxide or oxygen out of the stomata, because the oxygen released from the chloroplast would be provided to the mitochondria to perform the respiration, and the carbon dioxide released from the mitochondria would be provided to the chloroplast to perform the photosynthesis. Now there would be no enter or exit of carbon dioxide or oxygen. Have you got my point?
Yes, sir.
This point would be called as the compensation point, when the oxygen and carbon dioxide would compensate with each other. These would compensate with each other. So this is how we can say that during the night, the carbon dioxide increases. So coming back to a graph that is very important. I think you might have seen this graph already, but let's discuss about this. Just look at this diagram. This diagram is telling you that this graph is made from one midnight to another midnight. And you can easily observe here that during both of these midnights, the carbon dioxide level is decreasing during the noon. The reason is the photosynthesis rate is higher at this time, is higher. And once it is higher, the carbon dioxide level, because of getting used from during the photosynthesis, or you can say that because the plant is constantly consuming the carbon dioxide, it would be taken from the environment, and during the day, the level of the carbon dioxide would fall to 0.030. And during midnight, because there is no photosynthesis occurring and even the plants are respiring and giving away the carbon dioxide, so the level of carbon dioxide would be almost 0.040. You can say 0.043. It would be almost equal to this. The reason all of the organisms are respiring and the carbon dioxide level increases. So this is the variation that occurs around the 24-hour clock in the carbon dioxide. So what we were saying, we were managing to establish a point that light does variate. We can see it with our own eyes. The temperature does variate. We can feel it with the help of our body, the receptors of our body, but we cannot observe the. Yes, Zainab?
I had a question.
Sure.
So, at the nighttime, if every organism is respiring, how do we get oxygen?
Yes, this is a very good question. The answer is the level of carbon dioxide that is reaching at the highest point, the highest level of the carbon dioxide is 0.043. But we must know that the plant would perform so much photosynthesis that it would increase the level of oxygen during the day to 21% of air. This is actually the composition of air. For example, 78% of air is nitrogen. Oxygen is 21% and carbon dioxide is 0.03 at day and 0.04 at night. So the oxygen is at a very, very, very much higher rate as compared to the carbon dioxide even during the night. So it does not have any effect on our health. Got my point?
Yes, sir.
So, what I was talking about that we can feel the variation in temperature, we can feel the variation in light, but we cannot feel the variation of carbon dioxide around the day, around the 24-hour clock. So we perform the experiments. For example, this experiment, this graph that we have just discussed is actually a graph that has been made from a field. While carbon dioxide was added to the air from the respiration of all organisms in the field, we have taken these readings from a ground. If you observe this reading, if you try to get this type of reading in your room, you will never get this. Why? Because in your room, you are respiring. Some bacteria maybe are there respiring. Some animals are there, they are maybe respiring, but there is no plant here. So what does it mean? That in our rooms, there is not much variation. This is another answer to your question. In our room, there is not much variation in carbon dioxide as compared to a field. As compared to a field, there is not much variation. Let me put another question. We can say that the stomata closes at night. If stomata closes at night, then how does the carbon dioxide leave the leaf?
Through the cuticle.
Well done. It releases through the cuticle. It diffuses out actually because its gradient becomes so much higher inside the spongy mesophyll, inside the mesophyll tissue, that it diffuses out of the cuticle, because actually cuticle is fatty, and the carbon dioxide can easily cross the fatty layer, so it would be easier. So we have discussed this graph. Now coming to the graph that would be helping us on. Yes, this graph. It is very interesting. So right now we are going to discuss a graph about the limiting factors of the photosynthesis. Limiting factors of photosynthesis, and let's enlist those limiting factors once again so we can get the idea what we are studying. We are going to enlist the factor, one factor important CO2, the other factor is light, and the third factor would be the temperature. So let's put all these in the diagram. Number one, uh, while looking at the graph, you must know what is written at x-axis and y-axis. You must know. Because if you are reading one scale, then it would be a blunder, straightforward blunder. You would never get the proper reading if you are reading reading single scale, either x or y. You have to compare both of these. So increasing light intensity. So we have on x-axis increasing light intensity. First of all, observe this area. This area which I am right now shading is exclusively due to light. Why? First of all, the light is at x-axis. So if you shift this factor with the carbon dioxide, this area would be influenced by the carbon dioxide. If you shift this x-axis with the temperature, this area would be affected by the temperature. But right now in our current graph, we have light intensity at x-axis, which means that if we have no light, there is no benefit of giving the plant maximum carbon dioxide and maximum temperature. No, there is no benefit, because this area, this shaded area represents that if we have no light, there is no benefit. So first of all, establish this point that you must look upon the factor that is present at the x-axis. And without that factor, you are not going to uh, even start the photosynthesis. What does that mean? That actually means that mostly 98%, if you are asked this graph in the during your exams, there would be only light mentioned here, only light, because this is the importance of light. If you remove the light and you expect the plant to properly perform the photosynthesis properly, you give the plant maximum temperature and maximum concentration of carbon dioxide, but you do not have given the light, so this is useless. So after this, just observe the graph. Once you have, the graph has risen because of the increasing intensity of light, the light is increasing in towards the right. But just look at the graph that now it has become constant. It is not increasing further. Not increasing further. What is the reason? The reason is you have given 20 or 30 degrees centigrade to the plant, but you only have provided the plant with the 0.03% of the carbon dioxide, which although is a good level, not bad concentration, because this concentration is normal during the day, but still you have given it 0.03%. So it means you have uh, you are observing the effect of carbon dioxide. This is your focus right now. Actually, in the scientific language, this is known as the variable, independent variable, in which is you are changing. In There are two type of variables. Okay, let me tell you one more thing. In any of the experiments, because this would help you in further discussing the topic of experimental design. In any of the experiments, we have two, uh, or you may say, two types of variations. Two types of variations in readings. One that we can change on our own. On our own. Those readings are known as independent variations. These are known as independent variations, and the factor that we are controlling is known as independent variable. Independent variable. For example, if I am controlling the amount of carbon dioxide here, this amount is independent variation, and this factor is known as independent variable. That factor would be known as independent variable because controlling the I am controlling the amount of carbon dioxide. The second type of variation that occurs in the experiment is the dependent variable. Dependent variable. What is dependent variable? The dependent variable is the reading changes due to change in independent variable. The dependent variable is actually your result. It actually is your result in which your experiment is depending. You are during an experiment, when you want to prove something, you set yourself to derive a result. So once you derive a result, it would have some readings. For example, this is one reading of photosynthesis, this is the other reading of photosynthesis, and this is the other reading of photosynthesis. So right now, what do we have? We have the photosynthesis rate of photosynthesis as our dependent variable because it is getting changed due to independent variable. For example, I have a cup and I have water in it. I'm and then I have the sugar and I take 1/2 spoon of the sugar and put it inside the glass of water, cup of water, and then I drink it. I take one sip and I say, yes, this is sweet. Okay. Now I have taken what amount did I take first? I took 1/2 spoon. Now I took 1 whole spoon of the sugar and then I added it into the another glass of water. The amount of water is same, but I have put a whole spoon of the sugar and I put that whole spoon of sugar into another cup with the same amount of water and I drink it. I would say this is sweeter than the other one. Now tell me, what is the independent and what is the dependent variable in this experiment?
So the sweetness of water is the dependent variable, and the independent variables are water and sugar.
Now coming to the point. Independent variable is actually only the sugar because in an experiment, once we are this is another very important point which would help you in that topic. In an experiment, we must change only a single factor. At our level, we must change only a single factor. For example, let me tell you with the same experiment the effect of changing the two factors simultaneously. For example, you have taken a cup of water and you would put 1/2 spoon of sugar into that cup. Now you have taken a whole spoon of sugar and you put that whole spoon of sugar into 1/2 cup of water. That water with the 1/2 cup and whole spoon of sugar would be very much sweeter than the other cup. You have the same result here. But how would you conclude that did the amount of water made the result different or did the amount of sugar make the result different? What would you conclude?
We would keep the amount of water constant.
This is the point of the experiment at our level. not in the uh, you may say thesis level, MPhil level, BS level, because we can change. There are some other ways to change two factors simultaneously, but still you must know that at our level we must variate only single factor. So this is another important point, that one factor should be constant. So let me tell you. Okay, so it means that if you are giving the plant 30 degrees centigrade, then the temperature is constant right now. So no, do not assume the 30 degrees centigrade. Let's hide it. Let's hide it. You have given the plant the 20 degrees centigrade temperature and 0.03% of the carbon dioxide. And the temperature and the rate of photosynthesis suddenly got constant at this level. Now, just observe the other readings. What have you done? You have increased the amount of carbon dioxide 0.15%. From the 0.03% carbon dioxide, you have kept the temperature same. And at this level, the photosynthesis rate increases, but it still gets constant after a certain level. Now, what would you do? Now you are going to see that, uh, okay, if you further increase the amount of carbon dioxide, the rate of reaction does not exceed to this level. Further increase is useless. Further increase is useless. Why? Because you are providing the carbon dioxide with the sufficient amount of carbon dioxide, but you are not giving the optimum temperature. So let's move further. Now let's see that how does the temperature would affect the reaction. Just look at this. You have given the 30 degrees centigrade and compare the difference here. This difference and this difference. A huge difference between all of the readings. So it means that there are limiting factors and the factors that can alter the rate of reaction of photosynthesis. Understood?
Yes, sir.
All right. Mineral ions for growth. There are some other, another topic. There are two important minerals here. All right. Used for glucose. This was only this is mentioned here. Yes, only two are often discussed with the at this level. One of the minerals is the nitrogen, which is important in making the proteins. Obviously, nitrate ions or nitrogen, these are very important in making the proteins. But some other minerals are also mentioned: phosphate, potassium, and magnesium. The phosphorus is needed for the plant cells to make important compounds, including DNA. Potassium ions are required for enzymes in respiration and photosynthesis to work, and magnesium ions form a part of the chlorophyll molecule. The chlorophyll cannot be completed. Not only the part, the chlorophyll can never be completed without magnesium, which means no magnesium, no chlorophyll. So it is very important. Please excuse me for a minute.
Okay.
Okay. So any question?
No, sir.
We have completed this. Mineral, chlorophyll, ions, I don't know, some other one. Yes, we have completed this. What are we, where are we off to next?
We need to look at some of its practicals.
Oh, wow. Okay. What practicals are we aim to do? Investigate photosynthesis, showing the evolution of oxygen from a water plant and production of starch and the requirements of light, carbon dioxide, and chlorophyll. Oh, these are too much experiments. Hmm. Okay, we will be discussing in the next class, but let's me look. Start messages. Okay, yes. Plants and food, transport in plants, chemical coordination in plants. Okay, ecosystem and human influences. So, we need to prioritize the topics. Actually, we have less time. We need to prioritize the topics so that we can start the past papers. Let me finish the recording.